Hello students, this is Dr. Faizan Mirza. We are discussing homeostasis and the topic that I am discussing right now is excretion. Excretion is all about excreting the metabolic waste which are present in your blood plasma and they travel as a part of your plasma. When the plasma gets filtered in the kidney, the excretory products are sent to the urinary bladder to be sent out in urine. The metabolic wastes are the products of various chemical reactions occurring within the body, and these waste products are produced as a result of various anabolic and catabolic reactions inside the cells, and once they are produced they spill into the tissues and then into the tissue fluid and then into the blood. Major metabolic wastes, they include urea, uric acid and creatinine, and we'll restrict our discussion to these three primarily. Urea is formed in your liver. We'll be discussing the formation of all the three one after the other. Formation of urea. Urea is formed inside the liver by a process called deamination, and deamination occurs for all the excess amino acids which are present in your blood. The urea is formed in such a way that the amino acid loses two hydrogens and a water molecule is added. As a result, it turns to two products, number one keto acid and number two ammonia. Keto acid can then be respired or converted to glucose or fat, whereas ammonia gets added to carbon dioxide and the product is urea together with water. This urea then enters the blood plasma and is a major waste product in our bodies. The second major metabolic waste is uric acid. Uric acid, it forms by the breakdown of purine bases, which are adenine and guanine, which might be present in the mRNA or any kind of RNA and DNA. And once these bases are broken down, uric acid is formed. The third major waste product is creatinine, and creatinine forms from creatine phosphate. Liver produces a molecule called creatine, and creatine then combines with phosphate to form creatine phosphate, which is CRP. CRP or creatine phosphate is present in our muscles, where it provides a pathway for regenerating ATP during strenuous exercise. Creatine phosphate can donate its own inorganic phosphate to ADP, whereby the turnover of ATP is increased, and the increased ATP breakdown demand which is put on the muscles once you are exercising is provided by CRP as well. So what happens here? You know that ATP breaks down to ADP and PI to give you energy for workout or for respiration, for exercise, and respiration regenerates this respiration of glucose. So what happens with CRP? CRP, the phosphate group breaks from creatine and the phosphate group get added to ADP. ATP is formed, and this ATP can then be used to produce more energy or provide the cell with more energy. Lastly, what happens? We have the muscle breakdown can occur, and when muscle breakdown occurs, muscle breakdown causes the creatinine to form, and creatinine then comes into the blood plasma, and this is the third major waste product present in our blood that needs to be excreted out. This diagram here shows to you how the position of your kidneys in the lower back, how they are receiving the blood from the renal artery, which in turn is receiving blood from the aorta. This is the descending aorta sending the oxygenated blood to the kidneys through the renal artery. But this oxygenated blood is rich in the metabolic waste. So once the blood comes here, although the kidney cells are provided with oxygen, but the metabolic wastes are also removed and blood gets filtered. The filtered blood, which is deoxygenated now, having a low quantity of metabolic waste, is sent out through the renal vein and goes back to the heart through the vena cava. The filtered part forms urine. It travels from the kidney into the ureters. The ureters take this urine through peristalsis into the urinary bladder. At the end of the bladder, there is urethra, and there are sphincter muscles that prevent the leakage of urine, and you can void urine at your will when your bladder is full or when you want to void. This diagram shows to you the cut section of kidney. There are 4 layers in kidney. Number 1, capsule. The outermost layer is the capsule. Then the second layer is cortex. Third layer is medulla, and fourth layer is pelvis. Pelvis then joins with ureter. This is how a nephron is present inside a kidney. So cortex is having the Bowman's capsule, proximal tubule and the distal tubule parts of the nephron, whereas the loop of Henle and collecting ducts are present in the medulla. Collecting duct takes the urine to the pelvis, pelvis takes the urine to the ureter, ureter then takes the urine to the urinary bladder, and through the urethra, urine can be passed out of the body. This diagram shows you how urine formation occur. Urine formation I'll be discussing shortly in one of the videos. We'll see there that urine formation actually is a 3-step process. Initially, the glomerular filtration occurs where the filtrate is formed from the filtration of blood plasma. This is the Bowman's space into which the filtrate gets collected, and the filtrate travels, and from here the solutes get reabsorbed into the blood, whichever is required. This is called tubular reabsorption, and then tubular secretion occurs, which means some solutes are added into the filtrate according to the needs of the body. Parts of nephron. Nephron contain 2 basic parts. Number 1, the malphizian corpuscle, and number 2, the tubules. At times, collecting ducts are also considered to be a part of nephron. It depends on which book you are following. Malphizian corpuscle include 2 parts. Number 1, glomerulus, and number 2, the Bowman's capsule. Whereas there are 3 types of tubules present inside the nephron. These 3 tubules include your proximal convolute tubule, PCT, LOH, which is loop of Henle, and distal convolute tubule, which is DCT. Loop of Henle is then further divided into 4 parts. There is a thin descending segment, there is a hairpin bend, and then you have a thin ascending limb of loop of Henle, and finally there is a thick ascending limb of loop of Henle. So there are 4 parts of loop of Henle, and then there is collecting duct after the nephron. So these are the parts of nephron. And the blood vessels of the nephron include glomerulus, and apart from glomerulus, there are other blood vessels which include afferent arteriole, efferent arteriole, peritubular capillaries, and vasa recta. Where are these present? This is what we'll be discussing shortly. So if we draw a structure of a nephron, so this is how we see that the nephron is having a cup-shaped structure, which is joined to a coiled tubule. The coiled tubule then straightens out into a loop. The loop bends and travels up again into the cortex. Again, the tubule coils up and finally joins with a collecting duct. So this is how a nephron is present. The nephron parts include Bowman's capsule. Bowman's capsule is also called as the renal capsule. Bowman's capsule is having 2 layers. Number 1, the first layer is called as the visceral layer, which is in contact with the glomerulus, and the outer layer is called the parietal layer, which is in contact with the surrounding tissue of the cortex. The layer or the region between the 2 layers, which is visceral and parietal, is called as the Bowman's space or the renal space. This is where the filtrate gets collected. The first coiled tubule is called as proximal convoluted tubule or PCT. After proximal convoluted tubule, you have the descending limb of loop of Henle, the thin loop, and then thin ascending limb of loop of Henle, and then there's a thick ascending limb of loop of Henle. So these are the parts of the loop of Henle. And then you have another coiled tubule, which is called as the distal convoluted tubule or DCT. DCT then joins into collecting ducts, and you can see that the lining that I have just drawn in gray here, this represents epithelial cells of nephron, and all the cells are of single cell lining. This is the collecting duct, into which you can see multiple nephrons, distal tubules have been joined. So all these cells which are forming the lining of the nephron are all single cell linings. This is how the filtrate starts to travel inside the nephron. When the filtrate forms in the Bowman's space, so we have the filtrate initially forming here. The filtrate starts to move into the Bowman's capsule. From there it travels into the proximal tubule. The filtrate will then go to the loop of Henle. It will be transported along the loop of Henle and up towards the distal tubule. Once in the distal tubule, the filtrate will then be added up to the collecting duct, which will be receiving filtrate from various other distal tubules of different nephrons. The filtrate is now urine, and this will be taken to pelvis initially, renal pelvis. And from pelvis, it will go to the ureters, and from ureters, it would go to the urinary bladder. And this is how the filtrate flows. Now, if we try to link up all the associated structure of nephron with the blood vessels present, so this is how the blood vessels are present. It's an extensive branch network that we see all spread all over the nephron. And this includes various vessels, the ones that we already have listed. These are just the major ones. There are a lot more. There is a branch of afferent arteriole that is bringing blood to the glomerulus. Glomerulus is present right next to the visceral layer of the Bowman's capsule. And then there is an efferent arteriole, and then these are the peritubular capillaries, and here you have vasa recta. So afferent arteriole leads to glomerulus leads to efferent arteriole, and so on and so forth. You can see the change in the diameter of the afferent and the efferent. In normal status, efferent is having a lower diameter as compared to afferent. This increases the pressure in the glomerulus that promotes ultrafiltration. So from the renal artery, you receive blood into the afferent arteriole that goes into the glomerulus where ultrafiltration occurs. The unfiltered blood goes to the efferent arteriole, then travels all across the peritubular capillaries, and eventually this blood drains into the vasa recta and eventually goes to the renal vein. This is how blood flows across a nephron. The orange arrows here, they represent ultrafiltration occurring, whereas the brown arrows represent blood flow. So in nephron, you need to keep in mind there is a flow of blood across the blood vessels together with the flow of the filtrate inside the nephron. So there are two things flowing. Number one, filtrate, and number two, blood, or you can call number one blood and number two filtrate, and you have to keep both them with one another, separated by a single cell lining of the epithelial cells of the nephron and the single cell lining of these capillaries. This brings us to urine formation. Urine formation is a three-step process, and in these three steps, urine forms as the blood gets filtered and stuff gets reabsorbed from it and eventually secreted as well. So what's ultrafiltration? Ultrafiltration is about how the blood plasma filters out and the filtrate which is collected into the Bowman's space starts to travel along the nephron. You can see that the filtration occurs across glomerulus and the filtrate comes into the Bowman's space. All dissolved substances present in the plasma will become the part of the glomerular filtrate. This includes all the essential elements and ions and nutrients and vitamins and waste products, each and everything which is present in the Bowman's, which is present dissolved in plasma will be traveling into the Bowman's space, except for the large proteins which do not filter out, and the red blood cells. They stay back within the blood vessel and they go to the efferent arteriole. The major metabolic waste, which means urea and uric acid, you were supposed to just send these out. But apart from these, a lot of essential compounds also left blood and went into the filtrate. This brings us to the second step of urine formation, which is selective reabsorption or tubular reabsorption. This is about reabsorbing selected solutes which are required by the body back into the blood. As the filtrate travels along the proximal tubule and the loop of Henle and the distal tubule, these solutes are reabsorbed back and they are put back into the blood. For example, glucose and sodium and amino acid, inorganic phosphate, bicarbonate ion, magnesium, calcium, potassium, anything which is required by the body in the right amount will be reabsorbed from the nephron tubules into the blood vessels, which include the peritubular capillaries and the vasa recta. This is carried out by the process of active transport as well as passive transport along the epithelial cells in the nephron. The third step in urine formation is tubular secretion, and tubular secretion is about unwanted solutes which are secreted from the cells or from the blood into the filtrate. Excess of these are sent, for example, drug metabolites, for example, potassium ions or H plus ions or urea, and this ensures that the urine is more acidic as compared to the blood, and excess potassium ions are also sent out. So nephron tubules, they carry out the process of tubular secretions. So eventually this is all carried out by membrane proteins which are present in the epithelial cells of the nephron. So these are the three steps involved in urine formation. So displaying to you the structure of nephron once again, Bowman's capsule is dealing with ultrafiltration. Proximal tubule convoluted is involved with 67% of selective reabsorption. Loop of Henle deals with 25% of reabsorption, which include the reabsorption of water, sodium chloride, and urea secretion. The distal tubule is involved in pH adjustment and ionic adjustment, whereas collecting duct is impermeable to water, but in the presence of ADH, this becomes permeable to water. You can see how the nephron is divided into cortex and medulla. These parts, which include the Bowman's capsule, the proximal tubule, and the distal tubule, some part of collecting duct and loop of Henle are in cortex, whereas the entire length of collecting duct and the complete loop of Henle, it's in medulla. So this is how the nephron is divided, and this is what we saw in the diagram initially when we're discussing this structure in kidney, that these three parts are primarily inside the cortex, whereas the medulla is made up of loop of Henle and collecting ducts. Coming to kidney histology, you can see glomerulus here. This is the bunch of vessels, and this is the Bowman's capsule. This is the renal space here where the filtrate is collected, and right across it you can see this structure is the distal convolute tubule. It is cut along its width. You can see the lumen inside it and the cells that make up the distal tubule. Compare this to this structure. This is the proximal tubule. The proximal convolute tubule is a bit larger, and it is having brush border inside the lumen, and the cells are a bit more large in size as compared to the cells of distal tubule. If we look at the proximal tubule and we try to picture it, so the proximal tubule, convolute tubule cells are epithelial cells that have microvilli which are pointing towards the lumen. And when we look at it from the microscope, we see that it gives an appearance of a brush border. That's why we call it as brush border, which is seen inside the PCT, and PCT is the only part of nephron where the brush border can be seen using a microscope. And this is the filtrate traveling in the lumen, and for reabsorption to occur, it must cross these cells. These are the tubules cut along their width. So here you can see that these large ones, these are all collecting ducts. These thick walled ones, these are all collecting ducts. A collecting duct is like this, having large cells with bulging nuclei, and then there are smaller structures as well. So this is the thin loop of Henle, this is the thick loop of Henle, and this is the capillary. So there are various structures cut along their lengths. So all the parts which can be seen in this diagram or in this photomicrograph, they all differ in the size of lumen or the duct of or the tubule, and the diameter of the cell size, and the cell shape. That's it from my side. Thank you so much.